Hi, I'm Jonathan Lambert, a European Molecular Biology Organization postdoctoral fellow that works here at the Department of Biology at the University of Padua. Here in the Vallisneri building, in Professor Lucas Scarano's lab, I work with a team of researchers that investigates mitochondrial dynamics in human health and disease. Here in the Department of Biology, we have access to state-of-the-art microscopy, and in the Scarano lab we have developed fluorescent tags that allow us to image mitochondria. And in this image, you are looking at two cells where we have used a fluorescent probe to image the mitochondria specifically. So what are mitochondria? Well, mitochondria are cellular organelles which a surprising number of people whom we run into can proclaim, "Mitochondria? I remember studying those in grade school. Those are the powerhouse of the cell." This is in fact quite a true statement, as these organelles are where the endpoints of cellular metabolism take place. During this process, the food that we eat is broken down into smaller and smaller molecules, and during this catabolism, energy is released, which the mitochondria ultimately convert into a form of chemical energy that our cells can then use to perform all the functions we take for granted every day. Without mitochondria, our cells would not be able to generate enough energy to allow our heart cells to contract and relax, making our heart incapable of pumping blood and nutrients around our body. Without mitochondria, we would be unable to generate power to do some of our favorite things, like kite surfing or reeling in the big one for dinner. Mitochondrial dynamics is the study of the size, shape, and number of mitochondria. In this image, we are looking at a cell that is being observed by a transmission electron microscope. On the left of the screen, we see the entire cell. In the middle of the screen, we have zoomed in upon this image to observe a single mitochondria. Here you can clearly see the mitochondria has two membranes, which we call the outer and inner mitochondrial membrane. We then even further resolved the architecture of the inner mitochondrial membrane, which you can see in the image on the right. Here you can clearly see the inner mitochondrial membrane invaginates on itself and forms a unique structure called the mitochondrial cristae. These cristae are the energetic units of life, as this is where energy generation takes place. Our lab is currently exploring the interplay between metabolism and the architecture of the mitochondria themselves, as we are finding that the common phrase, "you are what you eat," applies to the mitochondria as well. Just as athletes eat protein to increase muscle mass, we are finding that when the metabolism of the cells change, their mitochondrial dynamics also change. In particular, we have found that when you starve cells by removing glucose from their diet, not only do mitochondria elongate, but mitochondrial cristae number increases as well. Using modern molecular biological techniques in proximity-based ligand labeling, we are now currently identifying the exact metabolic machinery that is responsible for these changes in mitochondrial architecture. As we find novel protein candidates, we are now making novel genetic knockout cell models so we can precisely identify how these proteins regulate the inner mitochondrial membrane architecture. Our research is of significant scientific interest because the maintenance of the architecture of the inner mitochondrial membrane is dysregulated during diseases such as cancers, neurodegenerative diseases, and heart attacks. Understanding how our diet and the actual food we eat regulates the shape and dynamics of the inner mitochondrial membrane may provide novel therapeutic targets for the treatment of these diseases, benefiting all humankind.